The Thylacocephala are a group of extinct probable mandibulate arthropods that have been considered by some researchers as having possible crustacean affinities. As a class, they have a short research history, having been erected in the early 80s. They typically possess a large, laterally flattened carapace that encompasses the entire body. The compound eyes tend to be large and bulbous, and occupy a frontal notch on the carapace. They possess 3 pairs of large raptorial limbs, and the abdomen bears a battery of small swimming limbs. Anictozoon is a fossil animal from the Silurian of Scotland. Originally described as an early chordate, recent studies suggest that it was in fact an arthropod. The Thylacocephala are bivalved arthropods with morphology exemplified by 3 pairs of long raptorial appendages and hypertrophied eyes. They have a worldwide distribution. A laterally compressed, shield-like carapace encloses the entire body, and often has an anterior rostrum notch complex and posterior rostrum. Its lateral surface can be externally ornamented, and evenly convex or with longitudinal ridges. Spherical or drop-shaped eyes are situated in the optic notches, and are often hypertrophied, filling the notches or forming a paired, frontal globular structure. No prominent abdominal features emerge from the carapace, and the cephalon is obscured. Even so, some authors have suggested the presence of 5 cephalic appendages, 3 of which could be the very long genticulate and chelate raptorials protruding beyond the ventral margin. Alternatively, these could originate from 3 anterior trunk segments. Adult specimens of Dolicharis measured over 30 centimeters in length. It sported a row of small appendages to assist in swimming, as well as 3 pairs of clawed segmented legs, but it was probably a poor swimmer, instead depending on ambush to hunt prey. It is known for its well-preserved, large compound eyes, sporting well-preserved individual retinula cells, which assisted in catching prey. Thylacocephala possess features which would suggest adaptations for swimming in dim light environments: a thin, non-mineralized carapace, well-developed rostral spines for possible buoyancy control in some species, a battery of pleopods for swimming, and large prominent eyes. This is supported by the Cretaceous species from Lebanon, which show adaptations for swimming and possibly schooling. The paleontologist Rolf provides many possibilities, but concludes a realistic mode of life is mesopelagic, by analogy with hyperiod amphipods. Further suggests floor-dwelling is also possible, and that the organism could rise to catch prey during the day and return to the sea floor at night. Like most other thylacocephalans, Clausicaris had a narrow flattened shield-like carapace, 3 pairs of large grasping limbs, and a battery of swimming appendages further back, along with absolutely enormous bulbous eyes. Based on this anatomy, it would have been a highly visual hunter, using its huge eyes to locate prey items and then snagging them with its long spiny limbs. And we're not even entirely sure what type of arthropods thylacocephalans actually were. They're generally thought to be some sort of crustacean, but their highly modified anatomy makes linking up their exact evolutionary affinities very difficult. Whatever they were, they must have been incredibly successful as a group because they first appeared in the early Cambrian and survived all the way into the late Cretaceous. It is universally accepted that the Thylacocephala are arthropods, yet the position within this phylum is debated. It had formerly been cautiously assumed that the class was a member of the Crustacea, but no conclusive proof exists. The strongest apomorphy aligning the class with other crustaceans is the carapace. As this feature has evolved independently numerous times within the Crustacea and other arthropods, it is not a very reliable pointer, and such evidence alone remains insufficient to align the class with the crustaceans. Of the features which could prove crustacean affinities, the arrangement of mouthparts would be the easiest to find in the Thylacocephala. The literature features some mention of such a head arrangement, but none definitive. Odoria alata from the Middle Cambrian Burgess Shale of the Canadian Rockies. Originally considered to be a crustacean, more recently it's been reclassified as a stemarthropod. Growing up to 15 centimeters in length, it had a pair of large eyes on short stalks, and may have had 2 or 3 smaller median eyes in between. A tube-like carapace covered most of its body, and the tail had 3 flukes for stabilization while swimming. It's also likely that Odoria primarily swam around upside down, based on the hydrodynamics of its body shape, similar to how modern horseshoe crabs get around. It has been suggested to have been an actively swimming filter feeder or predator, using its limbs to capture or sift food items from the water column, before using its appendages to transfer food to its mouth. Canidaspis was likely a benthic animal that lived walking along the seafloor. It had claws on the end of its appendages which may have been used to stir up sediment, or to scrape off the top layer, which Derek Briggs suggested may have been a nutritious layer of microbes. Large particles it stirred up would have been captured by spines on the inside of its legs. These spines would have directed the food particles to the organism's mouth, where it used its mandibles to grind larger particles. Its antennae served a sensory function. The spines on its head probably served to protect its vulnerable eyes from predators. Its limbs probably moved in a metachronal sequence to produce a rippling motion. Although Canidaspis probably did not swim, this could have helped propel the organism from under soft sediments. The appendages also produced currents which would have helped with feeding and respiration. Myriapoda was relatively large-sized arthropod for the era, with the largest specimen measuring up to 14 centimeters. They possessed a thick carapace covering the dorsal and lateral sides of the head. Their thorax was composed of 30 to 40 segments, each of them coming with a pair of legs. The shape of its armor made it so the legs had a very limited range of movement, barely being able to reach whatever was directly under them. The abdomen was composed of another 60 segments, finishing in a short telson with a pair of triangular flukes made up of 3 segments each. The genus was found to be non-monophyletic, with the 2 species found to be more closely related to other Hymenocarines than they were to each other. Euthycarcinida most likely engaged in a fast-paced nectonic lifestyle. Its large size means that it was safe from most of the other predatory fauna of its environment. Many features of the fossils evidence a free-swimming, pelagic lifestyle. Examples being the presence of a tripartite caudal rami, a feature only found in Hymenocarines, and that the carapace goes ventrally beyond the legs, which would have heavily impaired this arthropod's ability to crawl on the ocean floor. Its eyes also have a similar shape seen in modern pelagic crustaceans. What this arthropod ate has been a difficult question to answer due to the lack of cephalic appendages in the fossils. Modern-day arthropods of a similar size like lobsters, stomatopods, and giant isopods are mainly scavengers or predators. Tzoia would have grown up to about 20 centimeters long. They had large eyes on short stalks, a pair of simple antennae, a horizontal fluke-like tail fan, and 12 pairs of appendages along their body, with the front 2 pairs at the head end being significantly spinier, and most of these limbs also bearing paddle-like exopods. The large carapace enclosed most of the body, and was ornamented with protective spines and a net-like surface pattern that probably increased the strength of the relatively thin chitinous structure. They seem to have been active swimmers that probably cruised around just above the seafloor, with their stout legs suggesting they could also walk around if they flexed their valves open. The arrangement of their spiny front limbs wasn't suited to grabbing at fast-swimming prey, but instead may have been used to capture slower seafloor animals or to scavenge from carcasses. While historically considered deposit feeders, feeding by sifting through the sea bottom for edible organic particles, the 2018 re-examination considered Wapsha to have been an actively swimming predator of soft-bodied prey items, using its first 3 pairs of cephalothorax leg-like appendages to capture and manipulate prey, while moving its lamellated appendages in a rhythmic motion to propel itself through the water. Up and down movement of the abdomen and the tail fan was likely used to move vertically within the water column. It may have used the claws on its cephalothoracic leg-like appendages to occasionally rest on surfaces. Although it bears a remarkable resemblance to modern crustaceans, its taxonomic affinities were long unclear. A comprehensive redescription published in 2018 classified it a member of Hymenocarina within Mandibulata. Euthycarcinoids were a group of arthropods that lived between the mid-Cambrian and the mid-Triassic, but despite existing for over 250 million years their fossil record is incredibly sparse, and it's only within the last decade that they've been recognized as being close relatives of modern centipedes and millipedes. The earliest members of this group were marine, living in shallow tidal waters, but they quickly specialized into brackish and freshwater habitats and were even some of the very first animals to walk on land. Fossil trackways show they were amphibious, venturing out onto mudflats to feed on microbial mats, avoid aquatic predators, and possibly lay their eggs in a similar manner to modern horseshoe crabs. Most euthycarcinoid species are known from tropical and subtropical climates, but Antarcticarcinus hints that these arthropods were much more widespread and diverse than previously thought. Discovered in fossil deposits in the central transantarctic mountains of Antarctica, it lived in freshwater lakes during the early Permian, at a time when the region was in similar polar latitudes to today with a cold icy subarctic climate. About 9 centimeters long, it would have had a similar three-part body plan to other euthycarcinoids, with a head, a limb-bearing thorax, and a limbless abdomen ending in a tail spine, but its most distinctive feature was a pair of large wing-shaped projections on the sides of its carapace. These may have helped to stabilize its body when resting on soft muddy surfaces, spreading out its weight, or they might even have functioned as a hydrofoil generating lift while swimming. Pneumodesmus is a species of myriapod that lived during the late Wenlock epoch of the Silurian period around 428 million years ago. The fossil is important because its cuticle contains openings which are interpreted as spiracles, part of a gas exchange system that would only work in air. This makes it the earliest documented arthropod with a tracheal system, and among the first known oxygen-breathing animal on land. Myriapods have a single pair of antennae and, in most cases, simple eyes. A pair of mandibles lie inside the mouth. Myriapods breathe through spiracles that connect to a tracheal system similar to that of insects. There is a long tubular heart that extends through much of the body, but usually few, if any, blood vessels. Myriapods are most abundant in moist forests, where they fulfill an important role in breaking down decaying plant material, although a few live in grasslands, semi-arid habitats, or even deserts. A very small percentage of species are littoral. The majority are detritivorous, with the exception of centipedes, which are chiefly nocturnal predators. The process of adding new segments during post-embryonic growth is known as anamorphosis. During mating, male myriapods produce a packet of sperm or spermatophore, which they must transfer to the female externally. This process is often complex and highly developed. Arthropleura is a contender for the biggest arthropod of all time, and like with other arthropods of the Carboniferous period, this gigantism has been attributed to the greater abundance of oxygen in the atmosphere of this time. Many fossilized trackways have also been preserved, with some even showing Arthropleura moving around trees as it made its way through the Carboniferous forests. Because the mouth of Arthropleura has never been recovered, it has been difficult to say with certainty if it was a herbivore or carnivore. Instead, scientists have had to look at the digestive tract and coprolites of this animal, which have been found to contain spores from plants like ferns, something that is not seen in a carnivorous animal, suggesting it was a herbivore. It disappears from the fossil record during the early Permian, when the climate dried and the lush forests were replaced with arid and desert-like environments. Not only did the oxygen content become reduced from the lack of oxygen producing vegetation, the dry conditions would not have suited Arthropoda because of its crustacean ancestry. Crustaceans as a whole need moist conditions to stop themselves from drying out, which is why today you either see them in aquatic environments, or the shaded areas of the ground such as amongst the leaf litter where the ground cannot get dried out by the sun. Glomerus is a genus of pill millipedes that belongs to the family Glomeridae. The genus Glomerus is the largest within the family, comprising about 100 species along with a few hundred subspecies, varieties, forms or genetic aberrations. Pill millipede lives in leaf litter as well as in grass and under stones, with a preference for calcareous soils. In domestic gardens, they are most frequent along hedgerows and at the bases of old walls, where the mortar has started to crumble, leaching lime into the soil. It is less prone to desiccation than other millipedes and can be found in the open, even in sunny weather, although they are more active at night and prefer more humid areas. It feeds on old, rotting leaves, despite the higher nutrient content of freshly fallen leaves, and it can be responsible for recycling a significant proportion of the nutrients in the leaf litter. Juliformes millipedes possess long cylindrical bodies with sclerites, skeletal plates consisting of ventral sternites, lateral pleurites and dorsal tergites, fused into complete rings. They possess defensive repugnatorial glands on all body segments except the last few, and are the only known millipedes to produce quinones in their defensive secretions. Black millipede is most active from 1 hour after sunset to 1 hour before sunrise, although in summer it also becomes active in the afternoon. Like many millipedes, it coils itself into a spiral, with its legs on the inside and its head in the center, when it is threatened, but it can also flee with sidewinding movements. The genus Scolopendra contains many species of centipedes found across the world's tropics and warmer temperate areas. All Scolopendra species can deliver a painful bite, injecting venom through their forcipules, which are not fangs or other mouthparts, rather, these are modified legs on the first body segment. The venom of most species is not medically significant, however, bites from several species can cause intense and long-lasting pain and swelling. Mediterranean banded centipede is an opportunistic carnivore. It will attack and consume almost any animal that is not larger than itself. These include insects and small lizards. The species has alternating bands of black and yellow gold. At approximately 10 to 15 centimeters, it is one of the smallest species in the family Scolopendridae. Its venom is also not as toxic as that of other scolopendrid centipedes. House centipedes are incredibly fast and agile. They can move quickly with their long legs, enabling them to efficiently capture prey. These creatures are nocturnal and primarily hunt at night. They feed on other insects such as spiders, flies, moths and cockroaches, making them valuable predators for controlling insect populations in homes. They have venomous glands located at the base of their legs, but their venom is generally considered non-dangerous to humans. However, it can cause mild irritation or a burning sensation if it comes into contact with the skin or eyes. Contrary to their common name suggestion, house centipedes do not have 1,000 legs, but they do have numerous legs. In fact, they can have up to 15 pairs of legs, giving them a unique and distinctive appearance. In captivity, these creatures can live up to 7 years. However, their lifespan in the wild is likely shorter due to predators and environmental conditions. Despite their sometimes intimidating appearance, house centipedes are beneficial creatures that help control populations of nuisance insects in households. Remipedes are 1 to 4 centimeters long and comprise a head and an elongate trunk of up to 32 similar body segments. Pigmentation and eyes are absent. Biramous swimming appendages are laterally present on each segment. The animals swim on their backs and are generally slow-moving. They are the only known venomous crustaceans, and have fangs connected to secretory glands, which inject a combination of digestive enzymes and venom into their prey, but they also feed through filter feeding. Being hermaphrodites, the female pore is located on the 7th trunk segment and the male pore on the 14th. Remipedia have a generally primitive body plan compared to other extant crustaceans, and are the only extant pancrustaceans to lack significant postcephalic tagmosis. Because of the energy and nutrients required for swimming, molting, and to grow in size and length, it has been speculated that the larvae may have other sources of growth than its yolk, possibly symbiotic bacteria. Panacanthocera's was a member of an extinct group of crustaceans known as Kawasaki, close relatives of modern tadpole shrimp. Fossils have been found in Kazakhstan and northwest China. It was fairly big compared to most of its modern cousins, reaching at least 10 centimeters in length, and had distinctive spines around the edges of its carapace and its telson. It's not clear if it had eyes. There's a single opening near the front of its carapace that may have housed some, and so I've depicted it here with just one naupliar eye similar to the third eye of tadpole shrimp. It probably had a fairly similar lifestyle to its modern relatives, living in shallow freshwater and temporary pools and opportunistically feeding on everything from algae to smaller aquatic animals. The order Notostraca, containing the single-family Triopsidae, is a group of crustaceans known as tadpole shrimp. They have a broad, flat carapace, which conceals the head and bears a single pair of compound eyes. The abdomen is long, appears to be segmented and bears numerous pairs of flattened legs. The telson is flanked by a pair of long, thin caudal rami. Phenotypic plasticity within taxa makes species-level identification difficult, and is further compounded by variation in the mode of reproduction. They are omnivores living on the bottom of temporary pools and shallow lakes. The central autapomorphy of the Notostraca is the abandonment of filter feeding in open water, and the development of a benthic lifestyle in muddy waters, taking up food from particles of sediment and preying on small animals. A number of other characteristics are correlated with this change, including the increased size of the animal compared to its relatives, and the loss of the ability to hinge the carapace, although a central keel marks the former separation into two valves, the adductor muscle is missing. Water fleas are transparent, allowing researchers to observe internal organs and physiological processes under a microscope without the need for dissection. In freshwater ecosystems, Daphnia magna plays a crucial role as a primary consumer and is a key food source for many aquatic organisms, including fish and insects. Under favorable conditions, it can reproduce rapidly through parthenogenesis, where females produce offspring without fertilization by males. This reproductive strategy allows for explosive population growth. It is sensitive to changes in water quality, temperature, and pollutants. As a result, its population dynamics can serve as an indicator of environmental health and water quality in freshwater ecosystems. Due to its short generation time and ease of culturing, it is a model organism for studying evolutionary ecology, adaptation, and responses to environmental stressors in laboratory settings. Daphnia magna exhibits various defense mechanisms against predators, including rapid swimming movements, formation of protective helmets, and production of chemical deterrents. Research has shown that climate change can impact their physiology, behavior, and population dynamics, making them valuable subjects for studying the ecological consequences of global environmental changes.